was going to be the same.
Oh, good.
Was going to be the same day as Paul's birthday. And we also heard that over lockdown you'd gone a little bit crazy and started making your own bar. And we decided that you needed something in your bar to remember this project by. Cool. So every good bar needs a good sign.
Happy birthday to you.
Happy birthday to you.
Happy birthday to you.
Happy birthday to you.
Brilliant. That's just brilliant. Wow, cool. You breached my copyright though, Cormac.
I did.
Twice.
Yes, all over the place. But we can modify it to get it in a way that can be hung easily for you.
No, I know. I think I even know where to put it.
Yeah.
I like now with the bar, these little micro tasks keeps my brain busy.
All right, I'm gonna go.
Thanks, buddy.
No worries.
Irish people are really nice.
Well, yeah.
It was not just Irish people.
Yeah.
Well, maybe so, but an Irish person gave it to me.
Okay,
So he's
fair.
the nicest. So
Okay, let's get back to the business of the day.
okay, I stopped
so-
recording then.
No, you can continue to record if you want, please. And now you know how to edit, Sarah, so you can take out any nonsense.
Yeah, I know. Yeah.
Your Christmas project.
Thank you.
Okay, let's go to Vidya first. Vidya, do you mind if I spotlight you?
Yeah.
Trying to find you first. Vidya's a bit camera shy. I don't know why. She's very, very pretty.
No, I have my camera on today.
Ah, very good. You're getting used to it. Now, how do I spotlight you? Sarah, did you make me demote my powers on Zoom? Looks like you did. Can you make Vidya spotlight Vidya?
Yeah, I should remove you then. Video. Here you go.
Okay, so we have 5 minutes of questions for Vidya, maybe a bit longer, starting now. Who's first? I would be surprised if Walter Fowler didn't have a question, but whoever wants, just speak up.
I have a question.
Go ahead.
Here's Susanna. I don't know if I didn't get it very well, but I think it was on, I put it here, slide 5. So because you use this cell net thing to predict how well the models are similar to the different organs, right? And it actual cell net shows that RP-TEC seems to be really similar to kidney, but then from your assays, they seem comparing to your models, they actually seem to be worse.
Yeah.
How do you explain the differences?
Yeah, so the CellNet data, this is actually done by Maastricht University. And the CellNet data, it contains the information from cell lines, primary cells, and fetal kidney and everything. So it's a combination of all the data together. And we already know that RP-TEC, RP-TEC is from the primary cells, right? And it has a strong correlation, and you can see also that it has a strong correlation when they are grown on filters. So the model is getting constantly improved when you are improving the culture condition. And with the iPSC-derived PTL, we see that the similarity score is low to the kidney, but in some of the selected genes here, you can see a higher expression of proximal tubule marker, but this is some of the selected genes that are handpicked, right? So it's biased in
Okay.
And you can also see that some of the SLC transporter, for example, and some of the CYPs are absent in PTL. So it's not very pure, mature, I would say, but
Yeah, but this is the point. Like, is it better mature because it expresses the megaline complex, right? And shouldn't that be already like quite of a differentiated cell?
Yeah, so I mean, I would just to quote, like to quote Sarah's presentation. She had a quote saying that not all the models are perfect. I mean, you can use it for how they are performing, right? So that's why we focused here on maglin mediated toxicity because
Specific, yeah.
it seems to be functionally active.
So no offense, I think you answered the question well. Next question.
For me, yeah. Vidya, nice work. I have seen that you have had your cells in culture for at least 7 days. What would be, let's say, the max of the length of a good culture? Because you are claiming that you are doing experiments in repeated dose toxicity, and I think 7 days is a bit too short for that. So can you comment on that?
Yeah, so what the aim here was to test aminoglycoside mediated toxicity. So what was usually done is acute exposure for aminoglycoside, which they usually do it for 24 hours, and then and they overdump with aminoglycoside. For example, 10 millimolar of aminoglycoside. But what we try to do here is to perform what is happening in vivo, like they get constantly accumulated in the lysosome and endosomes. So that's what we try to do. So that's not acute and it's not long-term exposure, but it's kind of in between. So we stick to 7-day exposure, and for the model being used for repeated dose long-term exposure, we didn't test it yet for how long they can be stable. But we could see that they can be kept for long, but we just don't have a solid evidence that I can say that it can be kept for 10 days, for example. I cannot say that now, but we are.
Okay, again, Vidya and Baz, that really nice. I think you answered it well as well, and you can have follow up on Slacks. So anybody else? Last question for Vidya. If not, Baz, you can come back. Baz, continue then.
I have a question.
Yes, go ahead.
Thanks a lot for your work. I think it's very important to think also how, for instance, these models that you have been working on could be used for different applications. As you might know, we see now with SARS-CoV-2 infection, a lot of effects on the kidney. So basically a direct infection of brain with acute kidney injury and, let's say, proteinuria and hematuria. Do you think, because you said the model is as good as it is, do you have any idea, let's say, how your work could, and your model especially, could contribute to in vitro mechanistic research also related to SARS-CoV-2 infection? And the answer is correct, because I just an open question.
Yeah, so I'm not really up to date with SARS-CoV-2, but recently it was earlier it was known to be infected through ACE2, but I didn't update myself yet. And I have shown a slide where with the protein staining, I think slide number 6, where there is expression of ACE2, and yeah, and also we seem to have a induction of interferon signaling and such as immune response. So I think it's very likely that our model could be used for predicting those kind of to do those kind of testings. But yeah, so we need to we are still working on it. We are still working on testing other compounds. And
Vidya, in the interest of time, again, very, very good answer, very good question. We have to move on. Sarah, can somebody spotlight Sarah, please?
Thank you very much for all the questions.
Yeah, and please ask her more in the Slack. There's the Slack channel for that. Just go ahead if you have more questions. So Sarah, I gave a nice video on the blood-brain barrier. Who has a question for Sarah? Sarah, you have 5 minutes. It's always somebody has to start the ball rolling. Don't worry. I think people are fascinated by your talk. Nicoletta.
I have a question.
Okay. Go ahead, whoever that is.
So, Sarah, thanks for your presentation. Very nice. Did you check the type of genes that are regulated over the time in your control? So are they related? Because what do you think? Are they still differentiating or are they dying? I don't know.
So we've checked the genes, or at least the top 10 genes, and quite some of them are related to the extracellular matrix. So then I would think that they're more differentiating over time and not specifically dying in the control condition. And then I haven't performed, maybe I can perform some gene ontology to just have a more clear picture of all the genes that are changing, because there's still quite a decent amount of genes that are changing over these 15 days.
I think that would be interesting. Yes.
Thank you.
Anybody else has a question for Sarah?
Yes. Hey, Sarah.
Hi, Christian.
Question for Temposec. Two very quick ones for the Temposec.
Two questions. Hang on. A question.
Very quick. So the number of differential expressed genes through time, it looks really nice. First is just, is that all time points, or are there any other time points that are not on the map, that are not on the plot? And second, is this the differential expression compared to the treated chemical at day 0 or to the untreated chemical overall?
So first question, I only have these 5 time points that you've seen. So these are all the time points. And then if we're talking about the treatment and the untreated, so we're always compared to treated time points to the same time point of the untreated chemical. So in the presentation, you had first the part of the control where we compare with day 0 because we wanted to understand how much it's changing. And for the second part, where we're looking at the effect of the chemicals, we're really looking at the control of the same time point of that chemical.
Yeah, okay, so then only for the second part, I mean, I assume it would be the same to also look for the second part compared to the untreated, because whatever you take as a control, I think shouldn't, I mean, I think you should still get the signal for the chemical, but yeah, it's very, very nice.
Okay, thank you, Christian.
Thanks, Christian.
I liked it too. It was a really good one.
I don't know who's speaking now, so.
Thanks, Ivo.
Do we still have time or not?
Yes, we still have time.
Okay.
So there's 2 minutes left for questions.
Okay, so are you sure lactate is a good sign of stress for the endothelial cells? You use lactate release as a sign of stress, or?
I wonder where you got that idea.
What?
Yeah, yeah. So we use lactate as an indication of stress, indeed. But besides lactate, also the decrease in tear over time is indicating a stress on the barrier of the BBB. And I'm not sure.
And it is really paralleled by the lactate release.
Yeah, so the tear, we have measured the tear on more time points than we have measured the lactate. So we see the lactate increasing, but then in the meantime, for the highest concentration, we also had the problem that the cells are dying. So I think we need to take that into account for the cellular stress, that for the 50 micromolar concentration, it's not the very best reference, because we saw in the risazorin assay that we have a reduced signal, so probably the cells are dying. But I think for the 5 micromolar, it's a good reference, unless you know other downsides of it.
I don't know if you know that, if you know something about anything about the metabolism of these cells. So I don't know if they are increasing their glycolysis or.
I've been, yeah, that's a good question. Yeah,
I think that's something to follow up offline. Any more questions? Raise your hand. Actually, really, or do it with the way you can do it in Zoom.
Thanks a lot. It's really good to have a model of the BBB.
Thank you.
I think I don't see any more questions. Sarah, thank you very much. Who is the next person in the sequence of the videos? Sorry, I'll call that guy back. It's Shreya. No, it's Pranika. Sorry, Pranika. Am I right? It's Pranika?
Yeah.
Yeah, okay, Pranika. Pranika is very used to giving online talks, I guess. So go ahead, Pranika. Oh, no, What am I doing? You've already given your talk online. So questions for Pranika?
Is it so clear that nobody has a question now? Maximum.
Sorry, Tom, I'm in the middle of it.
You introduced a lot of things, including a comparison of the results of the TempoSec, and you presented, and definitely on purpose, the normalized count and the log change and the fold change.
Yes.
And this is something we have been struggling as well, because we were focusing on differentially expressed genes, and you participated in developing that. But anytime we were also told, and Thomas in particular was pointing at that, and he was right, that you should also take into account that maybe the fold change is high, but then the count is little. Now that you have played around, do you see anything better which would integrate the two information? Is there any way we can have one visualization only of these two kind of challenge, which is taking into account the degree of expression in the meantime as it changes?
I think the first thing, I hope I understood your question right, but sometimes you see the log fold change a lot, but the read counts which contribute to it has a lot of variance between the replicates which they represent. So I think it's very important that you calculate the variance or the standard deviation which is there in your read count, so that what you see in your log fold changes is actually a signal and not because of the variance you see in the replicates of for the same gene. So it's always important that you see, like you would have seen in the last slide of my presentation as well, that we have also plotted the normalized count and also the log fold change and also the variance, standard deviation, how it is. So you could see that there were some genes which have a lot of variance, even being the replicates from the same group, same condition, same dose, same time point. But I think like you have to really concentrate on such kind of genes before you decide that yes, they are really the ones which are the signals and not just contributed because of the variance in that. So I think plotting the variance would actually help, and standard deviation would really help. And in the log fold change also, you have this log fold change standard error, which is calculated by the DSEC2. And this is what you see in the plot as well for the log fold change. So you could also use that as a parameter.
Is there any more questions? We still have 4 minutes. Sandra, Sandra,
Okay.
go ahead.
Thank you so much. You can imagine that I enjoyed looking at your presentation because, let's say, quality of data is important to do the data interpretation. So, as you know, we have now issued the GIVIM. So we have about 62 reporting requirements that we would like to see when you're reporting on in vitro methods. From your point of view, looking at all the data, are there some, let's say, recommendations that you could give what you see as most important thing for the type of work that you did in terms of reporting of data? Any thoughts on that?
I would actually say the first and the second part of my presentation. So like how you organize the data is very, very important to have a very uniform, because I think in our project, we tried to do this from the beginning so that everything is uniform. And that's why we have even done the comparative analysis between different cell models. And the second thing is the quality control. So once you have the data, don't just start analyzing it, but do the quality check if it's really what you see is true, or it's just noise, or it's something random which is coming up, because not everything is a signal, even if it shows after the analysis. So I think these 2-3 things which I mentioned in my presentation are the minimalistic things which you should look at before you start analyzing your data, starting from the raw distribution of the counts, for example, in this particular thing. So I would say quality control would be the first most step for any kind of data, like be it TempoSec or any.
I'm looking forward to elaborate on that with you. Really great.
Yeah.
Very good. Is there any more questions? We have time for one at least. Marie?
Yes. Okay. Thanks a lot for your work. What you did on the data control and all the quality control is really great. Thanks a lot. So you said that you used cadmium for in the study, and you would like to see the temporal aspect of the response of the metal stress response and the oxidative stress response. So
Yeah.
I couldn't. Will you be able to conclude something about that? With
our main aim from that paper, like from that case study, was to see if we could actually resolve the temporal resolution of the mechanisms which are induced by the cadmium. So yes, we could, and that's why I couldn't put a lot of things there because of the time frame. But we see a very nice time resolution. We could group the different genes, and they showed same pattern. For example, in the metal response pathway, if you see in my slide, they show very similar expression, gene expression pattern over time. So that could like tell us that, okay, it started very early in the timeline and it stayed up until the end of the timeline. So this pathway is activated from the beginning to the end. That's what.
Okay. Veronica, thank you very much. Now,
thank you.
This is an experiment, right? Because it's really important. The ESRs. Shreya, I'm going to do you in the next phase. Sorry. Because I want to keep to the agenda. So somebody make the gallery back again, please. No? Okay, I manage it then. So what we're going to do is stick to the agenda. If you stick to an agenda, we can be creative. So it's free time now for 15 minutes, but don't go away just yet. So the plan is we'll just continue this as long as it needs, right? So I haven't uploaded all of the videos yet. There's a few missing, but don't worry about that. So in your free time, I will allow this time, permit you to watch the videos during your free time if you're not sitting down at your desk, right? So do not watch in the free time at your desk. You got to stand up, move around. The next videos are, somebody want to help me? I forget who's the next one we suggested, but it's
Shreya.
No, Shreya you should have watched that already, but if you haven't started with Shreya.
Okay, we start from Susanna and then my video.
Yeah, or just in the web page, watch the next one after that you've stopped with. And we'll meet back on the Zoom. I'm going to just leave the Zoom active. So if you want to discuss stuff or have breakout rooms, feel free. But I'll be back here officially at 12:20. So continue to watch the videos. If you have any questions, drop in or I'll use the Slack. So 12, isn't it? Yeah, 12:20 back here for question and answers.
Okay,
so,
I just wanted to also add that before watching Susanna's video, please watch the video from Ninke. And also watch the videos from Giovanni and Regina, and then.
Exactly. So always watch the PI before you watch the ESR, because that's a real nice link between them. And actually for Anya or for Cormac and Vidya, there's two PIs. So it's me and Anya. Watch Anya's is a very inspirational one. So watch that one as well. So good. So meet you back here at 12:20, or we'll just randomly chat with each other. Off you go.
Zara, I think you're now the host, so you have to activate the breakout rooms and add that people can choose. So under where you share the screen, you should have a button saying breakout rooms.
Yeah.
Click there, and then you will have some options.
Yeah.
And it says how many rooms you want to do. I mean, make 10. And then put that people can choose to go in and out.
Okay, and then assign manually, or let participants choose it.
Yeah.
Okay.
And then you can see who is inside the breakout room and enter if you decide to.
Yeah. So what I can also do is make Paul host.
All right, sorry, just 1 second. Will you stop the recording? So only record the group sessions. Sorry.
Just give me a second because I have to add me in Spotlight, remove someone else. And then, so who has questions, please raise your hand.
So it's 12:27. I'm still the moderator, Carolina.
Yeah, but I need to see, to add the spotlight. It's just that.
You're the video editor, that's true. So it's 12:27. We have 5 minutes for questions. Who's first? So Carolina, just remind us the title of your talk.
So I've been differentiating brain cells. So I talked about the brain spheres. And I'm SR5. Oh, Sarah. Okay. Yes.
Yeah, and I see Sandra also had a question. For me, the question was more about the paraquat results you presented. Is there, I saw the PCA and there were kind of two clouds of endothelial cells. The paraquat people, do you have any explanation for that?
So maybe I can also put Pranika, because she's the one doing, like, she is the one doing most of the scripts and really going deep in the data analysis. But indeed, so we try different things, and the final analysis is now going on, because we analyzed and we discussed and then we reanalyzed based on all the new things we discovered on the data. And because it's different study plans, we have to come to an agreement on what to do, because we have to do the same everywhere. So sometimes it's like we find something in one study plan, so we have to solve it and then go back to the others and do exactly the same. For the endothelial cells, we were having some questions, mostly in Zara's data, that we were trying to solve. So we hope that now it's closer to the reality, but indeed they are not.
And I,
yes?
No, I see Sarah has her hand up, but I thought Sandra, I can't see Sandra on my screen now. Oh, Sandra. So Sandra, you ask first, and then we go to Sarah afterwards.
Okay, so let me remove all the spotlight.
Okay.
And then,
do you hear me?
Yes.
Okay. So thanks a lot, Carolina. I really looked with a lot of enthusiasm to your registration, especially since you have been choosing also amiodarone as one of your test items, and I'm particularly interested in that because obviously, as I mentioned maybe yesterday, I'm coordinating this thyroid validation study with mechanistic methods, and let's say amiodarone is also important test item for thyroid dysfunction, both let's say hypo and hyperthyroidism. So I just wanted to know that you have an idea in terms of, let's say, the target cells in your human-induced pluripotent stem cells. What would you, if you would need to develop a method to look at thyroid disruption, how could you use your tool for that, to have some physiological relevant readouts related to that particular endpoint?
What we did TempoSeq, and in the list of TempoSeq, there are some genes that are linked to thyroid function and the pathways. So I think I texted you, and it's one thing we could do is to use the TempoSeq data and look particularly to the genes related to dysfunction.
Yeah.
And that could give us some hints, and then we could go further and see if these genes are really affected, and some others linked. So if you have an interest gene, it could be very easy for us to analyze it because we have the samples and so on. And indeed, when we look to the literature, we see in some papers that comes as amiodarone affecting brain function through the thyroid. So what they say is that first amiodarone affects the thyroid, and eventually it leads to some central nervous system dysfunction.
Just one quick add, if I can, Paul, but if you now are asked to make this as a throughput method for regulatory acceptance, so how would you go about that?
So the brain spheres...
For any compound.
But using the brain spheres, you mean?
Yes.
So the brain spheres in terms of high throughput, and what I know that you do in the GRC, could be a bit tricky because it's a 3D model, and it cannot be...
However, we need it.
Yes, it's great, but I don't really know how to use it with the robots you have and so on. So maybe we would have to do a screening in the 2D format and then move to the 3D and see all the cells together. I would, as you do it in the GRC. Maybe since I left, there were new things, and the robots are better because changing the medium is not as easy as in the 2D.
Carolina, I'm going to move it on. Thank you, Sandra. To Sarah.
Zara, can you raise your hand, please? I don't see you. Yes.
Just ask your question.
I also put myself, okay. So I just wanted to give an answer to Sarah's question about the PCA. I think the PCA that Carolina showed in the presentation is based on the raw data. So it's not the differentially expressed genes. So it just shows that how the raw data looks like. And I don't know if the percentages were there or not, but the highest percentage difference between them is about 17%, which is 1.7, which is really not that high. And also, I don't know if you could see it or not, that they are based on the control and treatment samples.
So, I couldn't see the difference in
yeah, okay. So maybe if you see, the question will be not there, because then we have the controls and we have treatments and the difference is not so. So I just wanted to give an explanation about it.
Okay. And indeed, when we were now doing the, to see if the different data we are using, they are all good, it improved a lot.
Okay.
So they came up as more distant in the beginning, and now we are able to get it a bit closer together.
And the paper is in advanced writing. So thank you, Carolina. It's Liadis now. Can you spotlight her?
Yes, just give me a second. And I need to...
So Liadis, can you just tell us the title of your talk so people can remember what it was?
Yes, my talk was about pig-glycoprotein modeling.
Okay, so any questions related to Liadis's work on pig-glycoprotein modeling? I don't see any hands going up. Well,
Nicoletta.
Oh, Nicoletta, very good, Nicoletta. Thank you. Go ahead.
Hi, Liadis.
Hi, Nicoletta.
I liked and I enjoyed a lot your talk, especially based on our collaboration on PGP. My question will be, well, it's not a question. I would like to know your opinion about the recent advancements of the DeepMind of Google's AI. But they recently published the AlphaFold that allows to build the 3D structure of a protein. And I was thinking how this will change the way the modeling, like homology modeling, the dynamic simulations that you showed, will be, what kind of impact this type of achievement will have in terms of working with the protein type of modeling.
I guess it's a really good achievement because it's going to allow us to have better models for the 3D structures of the proteins that are tricky to have experimentally.
Okay, yeah, thank you.
Thanks, Nicoletta. Any more questions?
I think Maxime has a question, but
yeah, Max, go ahead, Max.
Yeah. So yeah, I knew a bit of what you did, and PGP is one of our common things we like to play with, and it's known also to transport peptide. And I was wondering whether your approach would make it possible to see what interaction it can have with peptides, because as I remember it, you went for chemicals, including cyclosporine, which is slightly bigger, but peptide and A beta in particular is one target, is one substrate of pig-glycoprotein. Is it something you can expect to modelize? The interaction?
Sorry, I didn't get your question.
Can you modelize the interaction of A beta, the A beta peptide with pig-glycoprotein? Can you compute that, or is there any possibility to think about it?
I think so. I don't have really clear now the A beta peptide now.
Yeah, so A beta peptide is 40 amino acids. So it's definitely.
I think it's possible. It's possible to do the docking quite easily. For the molecular dynamic simulation, I think it's possible also, but it's going to take much more time to run one simulation because of the dimension, the number of atoms included.
Yeah. Because I'm kind of curious of it. I don't know whether there is any potential in doing it, but I doubt that anyone have any clue how this is possible, that the transporter is transporting peptide and which part of the peptide is interacting with it. I think nobody knows actually. So if in silico you can figure, it's already pretty interesting.
Max, is it known? Is the AB, what is it called again?
ABCB1.
No, am I getting confused? The Alzheimer's thingy.
What? A beta?
Oh, no, I got confused completely. Doesn't matter.
Yeah.
Any other questions?
Thomas.
Thomas Exner, is it? Yeah, Thomas.
Yeah, no, I just wanted to give a comment on that. I think docking will definitely not work because it's just too flexible. The 40 proteins, you can really dock very small peptides, but you end up probably with 3, 4, 5, something like that, not 40. For the MD, there is quite some literature out there, but it's also, you need huge amount of computer time to really do something useful.
That's a conversation you should continue.
Yeah, no, I'm out.
No, not complaining, Thomas. It's really interesting. And Dan Gierke is in the call as well. So you should continue that in a Slack channel and maybe have a breakout session, because that's really interesting, Max. Well, look, Lydia, to see what kind of interesting conversations you stimulated. So thank you very much for that. Now I wanna go to the next ESR, and that would be Nicoletta. I think there's an ESR student called Nicoletta, isn't there?
Nicoletta. Yeah.
So this is Nicoletta. Nicoletta, remind us of what your title was.
Modeling quantitative adverse outcome types of computational models.
Now, I'm sure many people will want to have questions, so let's do this in an orderly fashion. Who wants to be first? The first hand I see gets the spot.
I received actually a question on Slack, and I can elaborate.
No, you don't moderate your own questions. So that's so Susanna. Yeah, you go ahead, Susanna.
Okay. Okay, my question might be a bit stupid, but we'll see. Because I mean, since the beginning of the project, I'm still trying to understand exactly how practically Bayesian modeling works. And I'm still, so the question is a bit more, how actually do you work to actually derive probabilities for the data you find in literature? I'm not sure if this is a question that makes a lot of sense, but
it's a
practical
very
aspect
question.
of it.
I understand it. It was a pain initially for me. I think there are two ways mathematically to derive probabilistic models. And the one which you right now said to derive probabilities from literature, that's kind of going into Bayesian networks. The way I've presented with the presentation is not to take those decisions in that way, because maybe someone might say that those decisions are much more subjective. When you decide that it's 30% based on a test. So in the second, like the conceptual framework that we've built was based on giving a prior knowledge, but setting the type of distributions. So I can say that maybe my toxicity assessment, the EC values follow a normal distribution, and I want my model to evaluate against that type of distribution with specified mean, standard deviation, and whatever other parameters I'm interested in. So it's kind of giving a little bit more flexibility to the model, but it's about the mathematical way of defining the Bayesian model. And from my point of view, as well, there is actually a few books that also kind of try to set this distinction between the way you define the Bayesian model. And there are also packages on both sides, so it's up to us what we want to do.
Okay, to fit the data.
It follows actually this
Susanna.
conceptual, if I can.
No, go ahead, Nicola. You finish your thought process.
Just one more thing, because I know that it might be useful for Susanna, and I saw also her presentation. The way that Frederic Bouat told us at some point, like with PBBK, Bayesian model, that's the way I went. So it's not the other way that we can also find in the literature with setting the probabilities.
Indeed, that's the point, because sometimes they also use Bayesian for
Susanna,
PBK,
now your time is over, Susanna. It's Sandra's time. Sandra. Oh, you're muted, Sandra.
Okay. So for Carolina, I put something in the chat, and also for Nicoletta, so but I can say it myself. So more a little bit, let's say, in terms of the outreach of what you have been doing. So what do you see as the future of quantitative AOPs in AOP networks applied use more? And especially integration of the in vitro and in silico measurement methods for the key event relationships, because obviously we cannot have for each key event relationship a method So also there, what you're trying to do is to find, let's say, those critical nodes. We have a small example with skin sensitization, but I'm working on 18 methods. So how do you see your approach there helpful for what we are doing now?
Yes, thank you, Sandra. My personal opinion will be that as a QOP is data-driven, we need multidisciplinary team, and we need people that will sit together and will design the experiments, both the in silico side as well as in vitro. So we need a dialogue in that direction. And then in terms of how to prioritize things, those can also be done in a transparent way. One of the things of the many things that I've learned from Professor Mark Cronin was that whatever I'm doing, I need to keep transparency. So I need to make it clear what is being avoided and let our people, our scientists judge. And if there is a discussion and there is other types of agreements that can be adjusted. So the framework needs to be updatable, adjustable, but you need to build very reliable. Yes, exactly.
I'll take one more question. Thanks. Good question. Good answer. Is that Emilio? Are you trying to ask one? I'm not sure. Yes, Emilio. Somebody switch on. Emilio, unmute yourself.
Yeah, I was also writing that we will make available within Vega the tools for the molecular initiative answer that Nicoletta presented in her talk. One in the case of Liadis, the tool will be available this week. For the tools from Liadis will be available next year, let's say.
That's very good, Emilio, because it's a deliverable. So excellent. Okay, thank you, Nicoletta. We're going to unspotlight you now. And a break is coming up, so we won't take another ESR before the break. So I just wanted to tell you again, you can work during your break if you wish, or if not, don't. But make sure you don't sit in the same place. Can somebody unspot Nicoletta? How do you do that again? Oh, I should know this. Okay, I have it. So yeah, so we're going to have a break of 45 minutes. We're back here at 1:30, and maybe you'll catch another video on that.